Hello everyone, I am Rajiv Saxena, Associate Professor, Smriti College of Pharmaceutical Education, Indore. In today's video lecture, we'll discuss about the evaluation of disinfectant by Chick-Martin test, by Kelsey site test, and third is by use dilution test. In the last video, we have discussed about the evaluation of a disinfectant with the help of the Riedel-Walker test. In the Riedel-Walker test, the principle behind is the comparison or the evaluation of any unknown disinfectant as compared to that of the phenol. So at the end of the Riedel-Walker test, we get a value which is called as a Riedel-Walker coefficient, and if the value of the RW coefficient is greater than 1, then it is said that the unknown or the test disinfectant is having a better efficiency as compared to that of the phenol. And if the value is less than 1, then we can say that the test disinfectant is having the poor ability as compared to that of the phenol. So in continuation to that video, in today's lecture, we'll discuss about some other different methods which are commonly practiced in any microbiological laboratory for the evaluation of any unknown or any test disinfectant. So the first method is the Chick-Martin's test or the Chick-Martin's method. Now the Chick-Martin method or Chick-Martin test is actually having the similarity with that of the Riedel-Walker test. So it means here also we are comparing the efficiency of the unknown or the test disinfectant as compared to that of the phenol. But actually it was observed in 1908 by Sir Chick and Martin that there is some deficiency in the Riedel-Walker test, because in the Riedel-Walker test we are taking the test disinfectant in various dilutions or in various concentrations, and then we are adding the microorganism that is Salmonella typhi, and then we are calculating, we are evaluating that whether the test disinfectant at a particular concentration is able to kill the Salmonella typhi or not. But in that solution, we are taking only the disinfectant. But especially if we are talking about a hospital or any pharmaceutical industry, then what is actually our observation that in those places the presence of the organic matter is definitely available. If we are talking about a hospital, then what we can find that in the hospital some urine is also available, feces is also available, blood is also available. So if we are applying the disinfectant in those areas where the concentration of the organic matter is very high, in that case the disinfectant should have the capability to kill all the microorganisms even in the presence of the organic matter. So this point is actually raised by Sir Chick and Martin. So this Chick-Martin test is actually, we can say that it is a modification of the Riedel-Walker test. So the test is almost same as that of the Riedel-Walker, but what they have done, they have incorporated the human feces in the Riedel-Walker test. But further, it is observed that it is not very convenient to use the feces for the test. So further, Sir Garrod has replaced the feces with that of the yeast extract. So the current test which is we are considering as a Chick-Martin test is having the availability of the yeast extract or the dried yeast. It is actually the representative of the organic matter, and we have to perform the test, that is the evaluation of the disinfectant in the presence of the organic matter in the form of the yeast extract or the dried yeast. So as you can see the arrangement, here we have to take the different test tubes, and in these test tubes we have to keep the different concentration or different dilutions of the test disinfectant. Say for example, the highest dilution in the extreme right, again 1 is to 1300, then its lowest dilution, that is 1 is to 1200, then 1 is to 1100, then 1 is to 1000, and at the extreme right we have to keep the selected dilution of the phenol. So it is our general observation that 1% solution of the phenol seems to be very effective. So here at the extreme right, we have kept the selected dilution of the phenol, that is 1 is to 100. Then apart from the disinfectant, we have added the yeast, the dried yeast, and again the microorganism, that is Salmonella typhi. So after this arrangement, we have to keep all these tubes for 30 minutes at 20 degree centigrade. So it is actually the time period where the disinfectant and the Salmonella typhi, they are interacting with each other, and if your disinfectant is very active, then definitely it will kill Salmonella typhi. But that killing or that activity of disinfectant is actually taking place in the presence of the dry yeast, which is actually the representative of the organic matter. So after 30 minutes, what we have to do? We have to take out some of its sample from the first tube and inoculate into two tubes that consist of the growth media. Means here we have to test whether the disinfectant have killed the microorganism in the presence of the organic matter or not. So how we can check the activity of the disinfectant? Definitely we have to withdraw the sample from each and every tube and we have to inoculate that sample into the culture media, and if the culture media is showing the growth, definitely it means that disinfectant has not the ability to kill the Salmonella typhi in the presence of the organic matter. And if no growth is observed, then definitely we can say that the disinfectant is having the ability to kill the Salmonella typhi in the presence of the organic matter. So the presence of the growth or the absence of the growth can be observed when we take out the sample from these tubes after 30 minutes of incubation into the tubes in a duplicate that consists of the growth media or the culture broth. Then again after this, when the inoculation takes place, again we have to put these tubes for observation at 48 hours at 37 degrees centigrade for incubation. So after 48 hours we have to take out all the tubes from the incubator and we have to go for the observation of the growth. Again, if the growth is available, if you are saying it is a positive tube, then definitely we can say that in the presence of the organic matter at that particular concentration, the disinfectant is not having the ability to kill the microorganism. And if no growth is observed at a particular concentration, then definitely it is our observation or it is our result that our disinfectant at this particular concentration is having the ability to kill the microorganism even in the presence of the organic matter. So this test is actually called as the Chick-Martin test. Now, coming to the next test, which is also used for the evaluation of the disinfectant, it is actually called as the Kelsey-Sykes test. The another name of the Kelsey-Sykes test is the capacity test. It is also called as the capacity test. So as the name itself is suggesting that here we are trying to identify, we are trying to evaluate the capacity of any disinfectant, the capacity of killing the microorganism by a disinfectant at a particular concentration. Okay, so this can be understood in a better way by observing these particular figures. Okay, so in the first tube we have taken the bacterial suspension. So for performing the capacity test, first things that we need is the bacterial suspension, and for performing this Kelsey-Sykes test, different microorganisms are already recommended. Like for KS test, we have to go for the use of S. aureus or E. coli and Pseudomonas aeruginosa and P. vulgaris. So these are the different microorganisms that have to be tested for the capacity of a particular disinfectant. So in the first step, we have to take the bacterial suspension of any of this microorganism. Okay. Then what we have to do, we have to again take a tube that consists of the constant volume of a chosen percentage of a disinfectant. Okay. As you can see in this diagram, we have taken 3 ml of the disinfectant. So maybe I have prepared 3 different dilutions of a disinfectant as 1%, 1.5% and 2%. So initially first I have taken 1% of the disinfectant and I have kept the volume of the disinfectant as constant, and in this case I have taken 3 ml of the constant volume of the chosen percentage of the test disinfectant. Okay. So again what we have to do, we have to transfer 1 ml of this bacterial suspension into 3 ml of the disinfectant. Okay. Now this time, when the first time when we are adding the bacterial suspension into the test disinfectant, that time is actually considered as a 0 time or a 0 minute. Okay. So this is a time 0 when we are adding the bacterial suspension 1 ml into the 3 ml of the disinfectant. Okay. Then we have to wait for another 10 minutes. Okay. Means the bacterial suspension and the disinfectant will live together for at least 10 minutes. Okay. Then what we have to do? Now when the bacterial suspension is added into the test disinfectant, then at the 8th minute, what we have to do? Withdraw 0.2 ml from the tube and inoculate into 5 different test tubes that consist of the growth media. Means here we are actually taking the time period of the 10 minutes, but at the 8th minute what we have to do? We have to withdraw 0.02 ml of this solution of the disinfectant in which already we have added the bacterial suspension. Okay, then at the 8th minute we have to withdraw 0.02 ml and we have to inoculate into the first tube that consists of the growth media. The similar thing has to be done in all the 5 tubes. Okay, like as in case of the Chick Martin test, we have performed the test in a duplicate. So similarly, if you are talking about the KS test, here the observation of the presence or the absence of the microorganism is done for at least in 5 different test tubes. Okay, so what we have to do? We have to withdraw 0.02 ml at 8th minute and then inoculate into the media that consists of the culture broth. Okay, then again we have to wait for 2 minutes because at the 8th minute we have withdrawn 0.02 ml of the sample. Okay, then again we have to withdraw, we have to wait for 2 minutes. Okay, so when we wait for 2 minutes, so it is actually the time that is in our watch, it is actually 10 minutes because at the 0 time we have started the experiment. At the 8th minute we have withdrawn the sample from the disinfectant, then we have waited for 2 minutes. Okay, so the total time duration that is spared by the bacterial suspension with the test disinfectant is now 10 minutes. Okay, so when the time of 10 minutes is actually achieved, then again we have to go for addition of the bacterial suspension 1 ml in the same volume of 3 ml of the disinfectant. It means here we are not changing the dilution or the amount of the test disinfectant. In the same 3 ml at the 10th minute, the second loading of the bacterial suspension has to be done. Okay, because the name of the test is a capacity test. Here we are actually trying to identify the capacity of the disinfectant. Like if we are for the first time when we are adding the bacterial suspension, what is the effect? Then again we are again reloading, we are again adding some bacterial suspension and trying to identify the activity of the test disinfectant. Okay, so again and again we are checking the capacity of the test disinfectant. That's why we have kept the concentration of the test disinfectant as a constant 1. Okay, so at the 10th minute again we are transferring 1 ml of the bacterial suspension into the same 3 ml of the test disinfectant. Okay, then again we have to wait for 8 minutes and when 18th minute will be achieved, then again we have to withdraw 0.02 ml from this tube and inoculate into 5 fresh tubes that consist of the culture broth or the growth media. Okay, then again we have to wait for 2 minutes, then the time period will be, total time period span will be 20 minutes. Now we can say that the time period that is spared between the bacterial suspension and the test disinfectant is now, it is 20 minutes. Okay, so when the 20 minutes will be achieved, okay, when the 20 minutes will be achieved, then again we have to go for the addition of 1 ml of the bacterial suspension into the 3 ml of the same test disinfectant. Okay, it means at the time 0 we are checking the capacity of the test disinfectant, then at time 10 we are checking the capability of the disinfectant to kill the microorganism, and at time 20 again we are trying to observe the growth of the microorganism even in the presence of the disinfectant. Okay, so definitely at the 28th minute again we have to wait for another 8 minutes, and when the 28th minute will be achieved, then what we have to do? Again we have to withdraw 0.02 ml of this sample into 5 fresh tubes that consist of the growth media. Okay, so at 0 minute, at 10 minutes and at 20 minutes we have to load the bacterial suspension into the 3 ml of the disinfectant, and withdrawal has to be done at the 8th minute, at the 18th minute and at the 28th minute. Okay, so when the 28th minute inoculation will be done, what we have to do? We have to take out all these tubes and we have to kept it for incubation at 32 degrees centigrade for 48 hours. Again, we have to put all these tubes, that is 5 plus 5 plus 5, that is 15 tubes, and we have to keep all these tubes at 32 degrees centigrade for 48 hours of incubation. Okay, so after 48 hours, we have to withdraw, we have to take out all these 15 tubes from the incubator, and again we need to observe the presence of the growth in all these different tubes. Okay, so if the growth is available, we used to write it positive, and if the growth is not available, then we used to write it as a negative. Okay, so at the end, when we are withdrawing all the tubes from the incubator, we are checking the presence of the microorganism, and if the microorganism is available, we write plus, and if the microorganism is not available, then we used to write it as a -1. Okay, means no growth is available. Okay, so on that basis, we have to go for the preparation of a table. In this table, we have to write the concentration, because here first we have performed this test with 1%. Then the same test has to be performed with 1.5%. Then the same test has to be performed with 2%. Okay, so at different percentage, we have in the first column we have write the concentration. Okay, then this is the challenge number. First time when we are giving the challenge, that is at the 0 minute. Then second time when we are giving the challenge, that is at the 10th minute. The third time when we are giving the challenge, that is at the 28th minute. So this is actually the challenge number. Okay, so at the end, after 32 degrees of incubation for 48 hours, when we are withdrawing the tube and we are observing the presence and absence of the growth in the form of the plus and minuses. Okay, so this is actually the representation. Like when all it is showing the plus, means all the tubes at 1% concentration. In the first challenge also, all tubes are plus when the growth is available. Okay, in the second challenge also, the pluses are observed, it means again the growth is available. Okay, in the third challenge also, the growth is available. Then we can assume that in the 1% concentration, the capacity of the test disinfectant is not that much that it can kill the microorganism. Okay, so the similar observation has to be done for 1.5 minutes and for 2 minutes, or sorry, 1.5% concentration or for 2% concentration. Okay, so when more number of minuses are available, then we can say that it will passes the test, and if more pluses are available, then we can say that it will fail the test. Okay, so this test is actually called as the capacity test, or it is also called as a Kelsey Sykes test or KS test. Okay, clear? Now coming to the third test, which is called as a use dilution test. Okay, now we know that there are so many researches are actually taking place in case of the disinfectant. Okay, and in each and every day some new disinfectant is actually launching into the market. Okay, so every manufacturer before it's launching into the market claims that for the use of this particular disinfectant, this much concentration is necessary or required. Okay, so maybe for example, if you have purchased some disinfectant that is related to phenol, and they are claiming that if you are using this disinfectant, then definitely you have to go for the use of 1% of this particular disinfectant dilution. Okay, but for example, if I am having an industry, or if I am using this particular disinfectant in any hospital for a large scale disinfectant, so before using it, whatever the manufacturer that is claiming, first we need to check whether the manufacturer claims is correct or not. Okay, so for that checking, one more test is available, which is called as a use dilution test. Okay, this is again a very simple test. As you can see in this diagram, you can easily understand that what has to be done in the use dilution test. Okay, but the principle is very clear why we are performing this use dilution test, because every manufacturer, while launching a particular disinfectant, always claims that for this particular disinfectant, this much dilution is necessary. Okay, so before using this particular disinfectant in a very large manner in any hospital or any pharmaceutical industry, first we need to check whether this particular claimed dilution by the manufacturer is correct or not. That is actually the basis of the use dilution test. Okay, So now how we have to perform this use dilution test? Again, for the use dilution test, different microorganisms are already recommended, like S. aureus, Salmonella, Claraceus, Pseudomonas aeruginosa. These are some of the microorganisms which are recommended for performing the use dilution test. Okay. So what we have to do? We have to take this any one of the culture of this microorganism. Okay. Now in this culture, we have to go for dipping a metallic ring. Okay. As you can see in this diagram, we have dipped a metallic ring into the culture of any of these microorganisms which are recommended. Okay. Then we have to dip it. Then we have to withdraw that metallic ring from that bacterial suspension. Okay. Then after the withdrawal, this metallic ring is dried. Okay. In a very normal temperature or in a very room temperature, this ring is allowed to dry. Okay. So here we can conclude that whatever the microorganisms that are available in the culture, okay, they will stick on the surface of this metallic ring, and while we are withdrawing the metallic ring from the bacterial suspension in a dried form, at the end of the drying, these microorganisms will be available on the metallic ring. Okay. Now we have to put this metallic ring into the claimed dilution by the manufacturer. Okay, maybe he is claiming for 1%, maybe he is claiming for 1.5%, or maybe he is claiming for 2%. Okay, so whatever the claims are there, we have to prepare that particular concentration of the disinfectant, and we have to put that ring in which the microorganisms are already available in the dried form, and that ring has to be dipped inside that dilution. Okay, so here I have written dipped into the dilution of the disinfectant recommended by the manufacturer and left for 10 minutes at 20 degree centigrade. It means we have to put this metallic ring into the chosen dilution and keep that ring for 10 minutes at 20 degree centigrade. Okay, so it means the ring in which the microorganisms are available is having the chance to interact with the disinfectant, and if the claim by the manufacturer is correct, then definitely whatever the microorganisms that are available on the surface of the metallic ring will be killed. Okay, but we cannot see that whether the microorganism is survived or they have killed. We cannot see by our naked eyes. Okay, so how we can check that the growth is available or not? Definitely this can be checked by dipping that metallic ring into the culture broth that favors the growth of the microorganism. Okay, so next what we have to do, we have to withdraw this metallic ring from the dilution of the disinfectant, and we have to put this metallic ring inside the culture broth. Okay, then again I will read the following. This exposure, the carrier ring is transferred to a culture media and allowed the growth of any surviving microorganism. It means we are checking the growth of the microorganism on the metallic ring with the help of the culture media. Okay, so after the incubation, if the growth is available, then definitely what we can say that the claim that is done by the manufacturer is not correct. It means microorganisms are not killed at that particular dilution. Okay, and if no growth is observed after the incubation, then definitely we can say that the chosen dilution or the claimed dilution by the manufacturer is correct, and the microorganisms are having the lethal effect because of the disinfectant, chosen disinfectant. It means the claim by the manufacturer is correct. Now we can use this particular disinfectant in a larger scale, maybe in any pharmaceutical industry or in any hospital. Okay, so these are the different tests that are used for the evaluation of the disinfectant. The first test was the Riedel-Walker test. The second test is the Chick-Martin test, in which we are checking the disinfectant activity in the presence of the organic matter. The next test is the Kelsey Sykes test, in which we are trying to evaluate the capacity of a particular disinfectant at a particular concentration. Okay, whereas if we are talking about the use dilution test, here we are checking that whether the manufacturer claim of a particular dilution of any unknown disinfectant is correct or not. Okay, thank you so much.